Hello. In this video I'm going to be talking about or comparing mitosis with meiosis. So if you're looking at this video, you should already have a basic understanding of cell structure, a little bit about what a cell life cycle is about, and just some basic background in cell biology. Not in a very detailed way, but you know, if you're taking anatomy and physiology class, you know, you should have had lectures already on you know, the cell structure, cell function, cell replication, and that way this video will be more helpful to you because it will try to help clear up confusion you might have about these two processes, but I'm not going to be going into detailed points of each process. So it's important that you have a little bit of background. My name is Gary Senia. I'm a professor of biology at Suffolk County Community College here on Long Island. At the end of the video, I'll put my email up so that you can send me any questions you might have, or feel free to make any comments about the video. If you liked the video, you didn't like the video, all your comments are very much welcomed. Okay? All right, so just to kind of get started, right? A cell, now not all cells divide. Okay, so there is, you know, a topic of cells that do not divide, but that's really not part of this video. So I'm just going to be talking about cells that divide. So if we just confine our discussion to that point right now, a dividing cell, or cells that are capable of dividing, they're really doing one of two things. They're either dividing or they're in between divisions, in a broad sense. Okay. Now, what happens in between division? Well, that's a whole other discussion, and you know, it's a detail. There are detailed activities that go on in between divisions. But in a broad sense, a cell is either dividing or it's in between divisions. So keep that in mind. Now, when the first human cell becomes into existence, in other words, when an egg is fertilized by a sperm, right, you wind up with the first human cell, the first cell that will become the healthy adult human. Okay, that first cell is called the zygote. Okay, and the zygote has 46 chromosomes, has a full complement of chromosomes. Human beings have 46 chromosomes normally. Okay, so the first cell of the human has 46 chromosomes. Now, you should know at this point that that first cell, that zygote, is a combination of whatever was part of the egg and whatever was part of the sperm. So when the two kind of united to form this first cell, they contributed one half the parts, if you will, one half the material to each. They contributed one half each to the first cell of the human. All right, which means that the sperm and the egg would have had to have 23 chromosomes, not 46, because if each had 46 and they both contributed 46, well, then the first human cell, the zygote, would have 92, and that would not be a good thing. All human cells, with the exception of the sperm and egg, okay, have 46 chromosomes, right? So there must be a process, there must be a way whereby at some point cells can reduce the number of chromosomes so that during fertilization, when the egg and the sperm unite, there's that complement of 46 created again, okay? So that is very important for you to kind of keep in your mind at this point. Let's just kind of shift over a second here to this idea of cell division, purposes of cell division. Why do cells divide? Well, we know that the first human cell is called a zygote, and that an adult, okay, a fully mature, healthy adult, has many, many parts to them, right? I mean, we have many different tissue types, many different organs, each of which is made up of different cells. And these cells, okay, from that first cell to get to that very complex organism, requires a lot of change, a whole bunch of change that has happened between that first cell, okay, and the fully adult, healthy human, all right? A great deal of complexity has taken place. Okay. Now, what is the purpose, though, of cell division? Why can't just one cell just grow and grow and grow and grow? Well, cells, not all cells have the same lifespan. Some cells last for a few hours. Some cells last for a few years. Some cells last for decades. Cells die for various reasons. Cells die naturally. Cells die as a result of trauma. Okay, so cell division, or the reproduction, cell reproduction, replication of cells, is required to A, replace dead or dying cells. Okay, replace injured cells. Okay, and also just for general growth. Right? To get from that first cell to the fully mature adult requires a lot of growth, a lot of multiplication of cells, to create a multiplication of tissues, to create a multiplication of organs, to create organ systems, etc., etc. Okay, so cell division is very, very important. Now, as I started to say, cells are either dividing or they're in between divisions. So if we kind of just draw a timeline in a kind of a circular fashion, okay? Cells are doing a bunch of things, okay? And when they're not doing the bunch of things, they're dividing. So cell division is referred to as, or a process of cell division involves a specific process of mitosis, and mitosis specifically refers to the division of the nuclear division, okay, where the nucleus divides, okay. So when it's not in this process of mitosis, it's doing a bunch of other things, okay. So I could arbitrarily start anywhere, but I want to start where a cell has divided and formed two daughter cells. So let me just show you here for a second. Here we have a cell, right, with 46 chromosomes. It divides to form two daughter cells. That's what the D is for, two daughter cells, and each of those have 46 chromosomes. These two daughter cells will eventually divide to form daughter cells, which will all have 46 chromosomes. Okay. So, where I want to start is where there's a new cell that was just formed. In other words, the cell is divided, and now this cell is starting to grow. That's what happens when these two daughter cells are formed. The cell splits and forms two daughter cells. These two daughter cells are not the same size as the original daughter cell, but they'll grow to be that size. Okay, so that's kind of where we are right now. And that first time outside of the dividing time is referred to as G1. And G1 is kind of the point in a cell's life where it's just doing cell things, cellular things, normal cellular function, right? Which includes, by the way, kind of getting ready for the big event, cell division, nuclear division, okay? And part of this nuclear division, part of this getting ready for the cell dividing, is creating double the amount of DNA. Because when a cell divides, right, if the original cell has 46 chromosomes, chromosomes are what? Genetic material, DNA. If a cell has 46 chromosomes, when it divides, each daughter cell is to have 46 chromosomes, that means that DNA material must have replicated, must have doubled, so that when the cell split, each daughter cell would have an equal amount and end up with 46 and 46. Okay? Otherwise, if it didn't happen, and you split 46 chromosomes and you didn't double the genetic material, well, then you'd have half the amount in each daughter cell, and that wouldn't be a good thing. Okay? So part of this cell cycle is getting ready to divide. And part of that getting ready to divide time includes DNA replication. DNA replication. Okay. And that's referred to as the S phase. Maybe you can think of S for synthesis, making DNA synthesis, S phase. And then the next part, before it divides, is referred to as G2. And G2 is a phase where there's some tweaking of some proteins that are being made and a few other things. Centrioles are starting to duplicate. The cell is really getting ready for this main event division. Now all of this G1, S, and G2 is referred to as interphase. And then we have the main event, mitosis. Mitosis is looked at, is studied, I should say, in phases. And the phases correspond to certain positions that chromosomes take, certain characteristics of the nucleus at that particular point of study. We have prophase, metaphase, anaphase, telophase. Okay? These all require more detailed explanations, which if you refer to my video on mitosis, I go into more detail about them. But for now, let me just say that mitosis is a continuous process. Even though we look at mitosis as having phases, prophase, metaphase, anaphase, telophase, we can even be more specific and say early prophase, late prophase, early metaphase, etc. It's really one continuous process. Scientists look at these different phases because of certain characteristics that are important to study, but really mitosis is just one continuous process. And once the nucleus has divided, then the cell pretty much starts to divide, and the cytoplasm, there's cleavage in the cell membrane, the cytoplasm is getting ready to separate. And this separation of cytoplasm or division of cytoplasm kind of starts late anaphase and into telophase, and that's referred to as cytokinesis. Okay? It's really part of this mitosis process, all right? Even though it's talked about kind of like as a completely separate process. And in a lot of ways it is, because it involves the division of the cytoplasm, whereas mitosis is a process which describes the division of the nucleus. But there's a lot of overlap here, so keep that in mind. So now this is the general, scheme of life cycle of a cell and cell division, and all cells participate in this kind of, arrangement, with the exception of cells that don't divide, like I started out by saying that some cells do not divide. Okay? Now, meiosis is a special process whereby cells that normally would have 46 chromosomes can be reduced to have 23. And those cells that are necessary for that to take place are the germ cells. Germ cells are the cells that, germ cells are gametes, okay? Excuse me, I should say gamete is a germ cell, and, well, germ cell is a gamete. At any point, at any rate, it's a cell that will eventually become sperm or egg, so sperm or ova. There's precursors from the level of germ cell till you get to the sperm or ova that has the 23 chromosomes, and that process involves the process or the division steps known as meiosis. Now the germ cells, when they replicate themselves to become another germ cell, they, like any other cell, will follow mitosis. In other words, the process by which they replicate themselves involves mitosis. But the germ cells that will then become the sperm/ova, they have to go through a special division process. It's a two-step division process. Meiosis is a two-step division process whereby their chromosomes are reduced by half so that the end of the line cell, the sperm, ova, actually have 23 chromosomes. So that when they unite with their counterpart, when the sperm unites with the egg, they contribute 23 and 23 to form the first human cell that has 46. So this chart here kind of just highlights the differences. Are these the only differences between the two? No. Okay. So again, you should have a little bit of a background to understand some of the stuff that I've written here, but I'm going to just kind of broad stroke go over this kind of briefly. And if you do have a background, this will make a lot of sense to you. If you don't, you have to go back to your notes, go back to my other videos, and try to understand these two processes, try to understand the cell and why it divides and how it divides, and things that are important basic concepts. Okay, looking at the chart here, number of divisions for mitosis, what I'm referring to is one cell will divide once into two daughter cells. One cell, one division creates two daughter cells. One division creates two daughter cells. With meiosis, you have a two-step division process. It's called meiosis 1, meiosis 2. And it's actually in meiosis 1 where variation is created, which I'll talk about in a second. All right. Genetic shuffling. Now, by genetic shuffling, I mean the exchange of genetic material. None of that happens in mitosis. Okay? The daughter cells are identical to the original cell, which means they have the same amount and type of genetic material. With respect to meiosis, in meiosis 1, there's independent assortment and crossing over that takes place. Crossing over is part of a process called synapsis. And synapsis is, it's a little bit much more involved than the way I'm about to describe it, but for the purposes of this video, synapsis is like, if you could think about chromosomes snuggling up together. If, synapsis, snuggle, two S's, maybe that will help you remember it. But if chromosomes snuggle up together, they can exchange, parts of each other. There's a likelihood and an increased chance of them exchanging whatever's on one chromosome and the other chromosome. It's almost like when you snuggle up to somebody. The likelihood of whatever's on that person getting on you and you getting on that person is increased. Okay? And that's what takes place here. This synapsis crossing over. Genetic material is kind of exchanged, and it accounts for the genetic variability that we see in humans. In other words, although you have traits of your parents and your grandparents, there's physical characteristics that are similar. It's not, you don't have identical features. Okay, and the reason why you don't have identical features and you don't have you're not an exact replica of either parent is because of this genetic variation which takes place. Independent assortment and crossing over really ramps up genetic variability in humans. The result of each cell division. When one cell divides, okay, it forms two cells, okay, and each cell has 46 chromosomes. That's referred to as diploid, right? If the cell has 46 chromosomes, it's referred to as diploid, 23 from the mother, 23 from the father, maternal and paternal. It's referred to as being diploid. Okay, so one cell will form two diploid cells. With meiosis, you wind up with one germ cell, okay, that the chromosomes are reduced to 23, meaning that they don't have the 23 from the mother, 23 from the father. They're separated, and they have 23 by themselves, okay? That's referred to as being haploid. So one germ cell will end up with 4 haploid cells. And that's what I'm referring to here. Type of cell, with mitosis, generally involves somatic cells. When I say generally, because we know that germ cells, when they reproduce themselves to become another germ cell, well, they're doing that using the process of mitosis. Okay. Meiosis involves strictly sex cells. Genetic similarity, under mitosis you get identical cells, identical similarity. With meiosis, that's where you get variability, variation. Okay? So hopefully you found this video helpful. Again, I'm going to put my email at the bottom of this video. If you have comments or want to ask me questions, please feel free to do so. Thank you very much for watching this video.